welcome to the next presenter, and that is Andrei Osterman, who's a professor of bioinformatics and system biology, infectious and inflammatory disease center at Sanford Burnham Prebys Medical Discovery Institute. And he's also in this measuring the microbiome section, talking about genotype to phenotype, microbiome profiling methodology and applications. Welcome, Andrei. You're on camera, and I'll allow you to please share your screen.
Thanks, Ann. Yeah, so it's exciting to be in this meeting. So I'll present this somewhat more. Well, first of all, can you see my slide and can you hear my voice?
You are good to go. We can see and we can hear.
All right. Yes. So I'll present at least initially an academic aspect based on the work we're doing here in San Diego and Sanford Burnham Prebys Medical Institute, but I'll also come to some applications that are developing. My expertise and my long-term research interest over more than 2 decades with a number of colleagues is how basically to harness genomic revolution, especially methodology of reconstructing metabolic pathways and phenotypes from genomic data, and how to solve this problem in scale. So for a number of years, we were working in individual microbes from various types, commensal pathogens, environmental, and applying again reconstruction of various pathways and metabolic capabilities of these organisms based from genomic data and multiomics data. So the question when microbiome started, can we solve the problem of scale, and can we solve the complexity of microbial metabolic interaction in ecological niche using the same approaches that were proven effective for individual or for small groups of microbes? So essentially the talk will have 2 parts. I will briefly describe the approach that we're developing and the fundamentals of it without technical details, and the approach which we call the phenobiome. Basically, again, meaning the ability to predict metabolic phenotypes or other phenotypes from genomic data in microbial communities. And then I'll cover briefly in terms of what applications are in development. So again, just as a starting point of the layer, I will introduce the layer that we are aiming to add to the microbiome research. And again, obviously and I'm talking about, and there was a wonderful presentation that I was listening to on many good empirical approaches and models. So the question, can we supplement or support this developments of prebiotics therapeutic foods by still using better the power of genomics? And so what people are trying to do definitely is by using sequencing data that are becoming widely available to get to some sort of real life applications. And a typical layer that's used in the research and very productively is just to use taxonomic description of ecological niche, who is there and what are the numbers of various bacteria there, and also try to enumerate genes and maybe some pathways, trying to find association between changes in microbial communities and state of health and disease. So we believe, again, based on the experience with in silico metabolic reconstruction, that we can add another layer that can make this path from sequencing data to application more rational and more robust. Can we again just try to model and capture metabolic interactions and capabilities of microbiota using predicted metabolic phenotypes? And again, to the degree this kind of problem, consider obviously a problem of ecological niche, in this case being our guts and the microbial inhabitants there. And in obvious view, again, what we are doing on a daily basis is feeding various nutrients to this ecological niche. In large scale, we're feeding carbohydrates that are utilizable by bacteria, what we call typically prebiotics that are sources of carbon and energy. We're feeding amino acids in the form of proteins and peptides that are used both for catabolic purposes and also as building blocks for proteins by auxotrophs, smaller quantities, but very important as vitamins and cofactors that for some microbes are as important as for the host. So, and as a result, again, part of it is getting utilized by the host, but there's a lot of proceeds going to this community, which is utilizing it and actually exchanging different metabolites in various cross-feeding mechanisms and producing end products such as short-chain fatty acids that were mentioned a number of times, as well as neuroactive metabolites, which were also mentioned in the talk during the day. And again, the idea is that can we use rationally, not only empirically, or at least combining empirical studies and genomic studies to rationally and in a personalized way field microbiota-directed foods to improve the health. Again, this is obviously a very complex problem, but we're trying to make it ridiculously simple. I'm chemist by education. Chemists are simple. So again, the idea of the system, when you're feeding the nutrients in various forms, carbon sources, micronutrients, building blocks, and then they are spread between different microbial constituents that have different metabolic capabilities, different range of metabolic pathways that can either produce many of those from scratch, utilize from the diet, and also not only compete with each other for the niche, but also share and cross-feed metabolites within the niche, and obviously important interaction with the host. So again, this is a very primitive kind of description of the problem. We are thinking it's the first step. So again, the way we thought about this project early on, when we just started building, is essentially as a growing matrix. And the matrix that contains various microbes with sequenced genomes, bug 1, 2, 3, 4, 3000s, and phenotypes. And in this case, we are talking about very well-defined and measurable metabolic phenotype, such as ability to utilize particular carbohydrates, synthesize intermediary metabolites, produce end products. And these phenotypes in a simplistic way, again, before you get to expression data, can be captured as zeros and ones, present or absent. So the bug 1 can utilize this particular carbohydrate, cannot utilize this other carbohydrate, and can produce certain products. Again, very, very simple digital form, which would allow potentially, if accurate enough, to try to describe in a large way the phenotype of community as a whole and not just individual microbes, as we knew we could do. So again, to do that, we built essentially a workflow that was tested and developed in many aspects at the earlier stage. And we start again using a robust and well-developed reconstruction of metabolism from genomic data from integrated genomes in a particular database platform that we co-developed. And then, as a result of this reconstruction, combining all the data, develop this kind of matrix that I showed before, consisting of zeros and ones, representing metabolic capabilities and inabilities of inhabitants of this niche. So again, I'm not talking about what's under the hood. We spent many, many years with a number of colleagues to reconstruct metabolic pathways, and including the reconstruction of variants of this pathway. I'm showing just a small snapshot of utilization of particular carbohydrates that include transporters and downstream catabolic enzymes, and include variations between different groups of microbial species projected through genomic comparative analysis. But again, all this complexity is under the hood. Nobody wants to know that. But what is important that using this methodology, we can again capture the capabilities of any individual microbe in this very simple forms of zeros and ones. And that would describe the, for example, prototrophy, or ability to synthesize amino acids. In this case, this imaginary organism can make histidine and cysteine, but not tryptophan. The ability to synthesize vitamins. Again, in this case, this organism can be predicted to require vitamin B1 and B2, thiamine and riboflavin in diet, but not niacin, and so forth. Same goes to ability to utilize sugars and produce short-chain fatty acids. So in the current rendition of our reference collection, we have about 3,000 gut-representing microbes and more than 100 atomic metabolic phenotypes. Again, just one more technical notion that, of course, there is a lot of databases that capture individual pathways, but what's important to know that we focus on detailed reconstruction, which includes not only known canonical pathway for very well-studied model organisms, but also variations at the level of individual genes, like non-homologous gene performing same reactions, alternative metabolic routes, the same sugar fructose can be utilized by two completely distinct metabolic pathways, and not all of them are captured in CAG and other public domain integrations, and also taken into account transporters that drive prebiotic utilization phenotypes to a large extent. And without accounting for transporters, it's hard to be accurately predictive. Again, as a result, we're building this growing matrix of zeros and ones. This is still a small fragment of it, just to illustrate. Again, currently include more than 100 phenotypes for more than 3,000 human gut microbes. with complete genomes. And you see, again, without looking at details, the different groups of organisms possess wildly different metabolic capabilities. The variations are not that big with essential nutrients like vitamins and amino acids, although even here, very prominent members of microbial communities, for example, bifidobacteria, like in child's, have multiple oxytrophies when it comes to production of essential building blocks and vitamins. But of course, the major variations occur in the ability to utilize various monosaccharides, disaccharides, oligosaccharides, polysaccharides, and degradation of other carbon and nitrogen sources, as well as production of butyrate, propionate, and other short chain fatty acids. Actually, at this point, so this is already established, published, and used a number of times, but now we are expanding this space of genomes using metagenomically assembled genomes, which is a wonderful new technology using long tag bio reads. And then we can recognize real variability between the microbiome constituents at the level of strains, not only at the level of species, but strains. And you see in this small matrix is shown, that starts showing this white squares in addition to kind of brown or whatever, orange and green, and that reflects variations of a genotype within the same species. Again, people often do not appreciate sufficiently variations of phenotype even at the subspecies level between strains, and analyzing MAGs is helping to capture these variations. So again, going back to the workflow, what is entry is this binary phenotype matrix, or BPM, which is produced from genome analysis. Of course, we want to go to the next step and see how it can be applied. Now, this knowledge base, once it's built, to comparing various samples and between health, disease, and different dietary intervention, and predict and at least rationalize initially the changes in microbiota with respect to these major phenotypes, driving nutrient requirements, capabilities to utilize various substrates, and production of end products. Again, I will not go to the technical detail of the workflow, which is now fully automated, and only focus that on stating that the initial input for this analysis could be just enumeration of organs in the sample. It could be based on the PCR, on the qPCR, on 16S, shotgun metagenomic data. But one way or the other, everything ends up being a table, like two-column table of who is there and what is the relative abundance with various degree of precision. So what we do in addition to this kind of standard way to describe microbial community is defining various phenotypic or functional metrics. And they are different. Probably the simplest one and the easiest to understand and use is what we call a community phenotype index. In a sense, it reflects the fraction of the microbial community that is capable to implement a particular metabolic property. For example, producing butyrate, or utilize xylo oligosaccharides, the polysaccharides. And it can be simply expressed in percentage, and you can get this kind of global metric of the community population, and then look how these capabilities are changing as a result of either dietary interference or in conjunction with different pathological or healthy states. Again, just to show very briefly that there is no magic there, and there is a really rational path from knowledge of pathways. In this case, I'm showing the example in B vitamin metabolism, metabolism of B vitamins and related essential cofactors, again, which we captured in genomic database. Based on the analysis of these pathways with all their nuances and different reference organisms, we're developing a simple pathway signatures or pathway rules that in the end allow in a very simplified way to go from genomic signature to ability or inability to produce, for example, in this case, thiamine pyrophosphate through the de novo synthesis of thiamine or B1. Again, once we generate this indices for all representative organisms, you get, again, this fragment of a matrix with the zeros and ones, and you curate it, and that's what we published a couple of years back. Then we can look how the representation of this community phenotype indices goes in a large number of samples, because the pipeline is producing these numbers automatically. And for example, when we analyze a number of large data sets, this example is from American Gut Product, subset, a few thousand samples, you see that capability to produce vitamins B1, B2, B3 is varies differently between people. That's not surprising, but what's also important that it's rarely 100%, which suggests that many inhabitants of our guts actually are oxytroph for major vitamins, and they will not be able to survive unless they get it supplied from the diet or from their neighbors. In fact, since we know that most of the water-soluble vitamins in normal diet are being effectively absorbed in the upper intestine, that came up the hypothesis that probably the exchange of vitamins between prototrophs and oxytroph producers and consumers within the community may be an important factor stabilizing the microbial community, to the point that it may not be very sensitive to the presence or absence of vitamins in the diet from purely microbiome perspective. We were pretty surprised that when the experiment was done in autobiotic mice colonized with a normal human microbiome or other mixture of those, what we did is indeed saw in the experiment that underdosing or depletion of vitamins from the diet or overdosing of vitamins in a diet given to mice in water actually did not affect the percentage or representation of oxytroph in these microbial communities, which again supported the main hypothesis that vitamin exchange, micronutrient exchange, is robust enough to support stability of the community well in the face of significant dietary changes. Again, that's to be said about the normal microbiome. Of course, if the representation of oxytroph is initially very high, which happens in many individuals, of course, the conclusion might be better, and some of the vitamins may be actually very important therapeutic food additives. But at least for the normal microbiome, the homeostasis, it's what's observed and is driven by metabolic capabilities of microbes themselves. Another, now kind of moving a little bit to potential application, I'm just showing the example from the application that may be used for diagnostic or sample classification, patient classification. So for example, we use the public data set of 16S sequencing for healthy patients and patients with Crohn's disease, inflammatory bowel disease. And of course, they have somewhat different taxonomic signatures and they were published, it is known. But we also used our approach and look what are the phenotype level differences between people carriers of the disease versus healthy controls. And again, with all the variations in the data, and I appreciate what was mentioned by the previous speakers that of course comparing a sample to other between people is challenging, and longitudinal sample of course is the way. But in this case, even this rough comparison suggested that patients with Crohn's disease on average have a significantly lower production capability for butyrate and higher production for propionate. Again, that's consistent with microbial signatures, but I want to emphasize that there is an interesting actional aspect in this type of sample classification. You can at least speculate that maybe increasing butyrate production, not necessarily through delivery of butyrate, but through promoting the toxin that can produce it, that may be causative and that may help to deal with the disease and go beyond diagnostics. But clearly the correlation and kind of actional phenotype differences are observed. I give also a couple examples more of our collaborative studies with a wonderful group of Jeff Gordon in Washington University. We worked with this group for a number of years, essentially doing the bioinformatics analysis associated with many preclinical and now clinical studies. So this example from a couple of years back, when together with Jeff Gordon team, we're looking at microbiota development in children, and especially in this case in children with severe acute malnutrition, which are significantly impairing growth. It's not only a problem of food insecurity, but also a problem of immature or incorrect development of gut microbiota starting from day 0 up to 2 years. So what's shown in this heat map, and again, I understand, I'm not meaning to show the details, that there is significant restructuring of microbiota phenotypic capabilities, which are reflected here by individual phenotypes going through this 36 months of development. And again, if this development doesn't happen timely or correctly, then there are severe health consequences. Again, one of the ideas of the study was to use this way of measuring microbiome to support or development of microbiota targeting foods. In this case, of course, in this work supported by Bill Gates Foundation, the challenge is that you need to operate with locally available foods and not with some exotic ingredients. And this work actually now in a phase of clinical trials, and there seem to be a lot of good progress by people who are doing the studies, in this case in Bangladesh. So the other example is probably closer to home, is not children, is adults, and these are people with obesity. And again, of course, everybody is aware of Jeff Gordon's seminal work about interaction between the host and microbiome as related to obesity syndromes. And in this study, which was published recently in Nature, we were monitoring, again, in our group, we were monitoring changing in the microbiota phenotypic capabilities, metabolic capabilities, as a result of fiber snacks, feeding. And that was done in 2 arms, both in mice, germ-free mice colonized with human microbiota, and also in obese people. And again, with all the, of course, variations and noise that present in the study, we were delighted to see that when we use the approach that I just described and look in 2 different ways through to the outcomes, we could observe that pathway enrichment analysis based on shotgun metagenomic data or 16S profiling pretty much converge to the same conclusion as to what are the driving phenotypes in the changes of microbiota as a result of fiber feeding. And in both methods, actually, again, as a coincidence to the, maybe not a coincidence to the previous presentation, arabino oligosaccharides and xylulose oligosaccharides, probably arabino xyloglycans, were actually major phenotypes that expanded in the microbiota of those patients or those mice that were provided fiber snacks, which clearly contained this type of oligosaccharides. Again, this is just to illustrate there is a possibility with all the caveats and all the limitation of the approaches, just to use genomic data to identify driving phenotypes and driving nutrients in this type of diet intervention studies, which again, hopefully will be very useful for people for people like previous presenters developing novel prebiotics. And in the last couple of minutes, I'll just say that, again, I presented more like an academic aspect of our development. And I'm delighted that a company which was founded a few years ago, I'm one of the co-founders. The company was named Phenobiome, based on the approach that we're using, is kind of applying this methodology in a number of collaborative studies, also targeting development of therapeutic foods and diagnostics. So I just want to show the example of the sample report which produced by Phenobiome. And the difference of the approach that is using in this assay is that in addition to, again, taxonomic or phylogenetic description of community, the sample, in this case, fecal sample, is analyzed and runs through the Phenobiome profile pipeline and presents the results of the analysis, not only in terms of what these microbes that colonize a gut of this person, but also what are their metabolic capabilities. For example, in this case, individual is shown that butyrate production of this individual in comparison to a reference group, healthy control, is kind of in a normal zone, is a little bit less than the mean, but clearly not in the underrepresented area. There's about 30% of community that is capable to produce butyrate. And I understand capable doesn't mean producing, but that's a first step. And these are the main bacteria, Faecalibacterium, Lactobacillus and generally that contribute to production of this particular short-chain fatty acid. And a similar analysis is performed with respect to sugar utilization capabilities, again, kind of in a normal zone for this particular patient or a particular person. And these are the main drivers of the fructooligosaccharides utilization, same about vitamin production. You can note that this person is kind of low in microbes capable of producing pyridoxine. Doesn't necessarily mean it's a bad thing. At this point, it's just an observation point. And you can probably say that the reason why this person is low in production of pyridoxine, because the bacteroides that are main drivers of these phenotypes, actually, they're not out of range, but in the lower side of the range between 4 and 40%. So if potentially that was a concern, which is probably it isn't, there would be a clear guidelines as to what probiotics or what prebiotics you should deliver to this person to enhance this particular phenotype. again, this is a sake of example, not a practical case in this particular phenotype. Same with amino acid production. And again, the other aspect of the report that Phenobiome is providing to practitioners is, again, the potential. It's not a recommendation, but at least what's called a nutrient. Phenobiome is calling nutrient map. So which shows which nutrients, which specific food additives or therapeutic foods can be used to, for example, enhance representation of these underrepresented species. Right. So, for example, if bacteroides would be considered underrepresented, you can use acetylglucosamine containing oligosaccharides. Well, some of the substrates are trivial, other can be more specific. I really appreciated the notion of specific and non-specific prebiotics. It's very close, very dear to our approach and very close. And how you can do it also to affect the more bacteria that are underrepresented without necessarily upsetting those that are in range and also important for the healthy state of the gut. So I think I'll stop here, acknowledging my colleagues in the lab, obviously collaboration with Jeff Gordon lab, which already delivered a lot of very interesting results, and collaboration with a Phenobiome Incorporated that is kind of commercializing some of the applications.